Welcome back, everybody. Nice to see lots of familiar faces. A lot of people here, if not everybody, would be very familiar with what we're going through today. And really, it's a lot of information, of course, to take in. And there's a reason why we refresh it every year. And even after that, there is no there's a responsibility on you, of course, but the expectation is that if you're in doubt, that you will clarify it with whoever is in the lab with you at that point in time, or if it's a project-related query with regard to hazards, that you will query it and be sure and with your supervisor. And that's important, okay? It's really important. So Richie's gone through the chemical waste disposal. I'm going to briefly go through the biology and biological waste disposal. And Richie mentioned something very important, of course, washing your hands before you leave the lab. But one thing that we're noticing, and it's not really it's not good practice, and we just want to avoid it in the future, is we're all back together now at the start of a semester, and you're going to find a surge in head colds, coughs, colds, flus, all of that sort of a thing. It's just nature. People get together, clustered, and infection can happen. People will tend to reach for the nearest hand towel that's close to them in a lab in order to blow their nose. You know, I get it. Your nose is running, you need to blow your nose. But always use the hand towels that are actually at the sink, okay? That's really, really important. Never use hand towels that are scattered or dispersed throughout a lab, because they have been used for practical purposes. They are there for experimental reasons. So they are possibly contaminated, and it's the worst thing you could do. God knows what you're going to rub against your nose, okay? So just bear that in mind. We're not going to be annoyed with you up and down grabbing a bunch of tissues, but grab the right ones, the ones that are for hygiene purposes, and not those ones that are scattered throughout the lab, okay? So just very, very important. The other thing is that for biological waste, it's important that you have to realise that you may need to wash your hands repeatedly throughout a lab. It's not just as you're leaving the lab, okay? So if you're handling biological cultures, live cultures, and you've carried out a transfer of liquids, you don't necessarily need gloves for aseptic technique. But it might be just good practice to wash your hands continuously throughout a lab like that, because of the nature of the culture that you're working with. So bear that in mind in addition to leaving the lab. Phones are great. They have super cameras on them nowadays. In fact, sometimes they can take much better imagery of protein gels and so on and so forth than the camera apparatus that we might use. And so for various reasons, you might be permitted to use your phone in a lab. But unless you're permitted for a particular purpose, you cannot have your phone out in a lab. It's just not safe, okay? Because you may get a reagent splashing on it by somebody else. You could have been keeping an eye on your phone and somebody else might splash on it. It could be acidic, it could be anything else, or it could be culture. So unless you're using it for the intended purpose, keep the phone away. If you need or are waiting on an emergency call, we're okay with that. That's not a problem. Remove your gloves and take the call if and when it happens, okay? Very important. But if after you're using a phone for a particular experimental purpose, when you're leaving the lab, it's important. We recommend that you take some of these tissues here and use a bit of the disinfectant that I'm going to describe later to just wipe down the surface of your phone and to make sure that it's clear for you to use, because you don't want residue building up on it, and if there's contamination on it, it's on your hand, it's on your face, and so on, okay? Okay, the Virkon disinfectant that we have in our labs is pink, and it's bright pink when it's fresh, and it fades when it's not fresh. So you are at nothing using that disinfectant if it's fading in colour. It's also pleasantly smelling when it's fresh, and it doesn't smell so great when it's losing its efficacy. So in fourth year now, the responsibility is that you will automatically clean down benches regardless of what you've been doing. It's just good practice before you leave the lab, and then during something like an applied decontamination, maybe because you've spilled a culture and so on, you need to make sure that you have the freshest Virkon there. As I said earlier, the technicians will have lots of backup if it's not looking as fresh as it could be, and on an occasion there may have been a container of Virkon that was not refreshed and unknowingly, and so don't be afraid to ask the technicians for fresh Virkon. It's readily available, okay? So second nature to clean down your bench space before you leave. Keep gloves on. This is not to come into contact with your skin continuously, because it is there for a reason. It's got disinfectant purposes, so the reagents that are in it are quite hazardous, okay? Well, can be. Richie also mentioned the autoclave. So pretty much I'd say everybody in this room will be involved probably in a practical project at some stage in semester 2. Obviously, yes, use gloves because the materials will be at 137 degrees when it's finished, but also do not put the lid on backwards, because you'll crack the seal on the autoclave, and that it's not going to autoclave for you, and also that autoclave has to be maintained and fixed, so it's out of circulation, and there's a big pressure on people then for project materials. But in addition, when, as you all know, pressure rises in an autoclave, the valve will lift. So even though your cycle is finished, the pressure won't have reduced immediately in that autoclave. So the valve will stay lifted and will drop when the pressure comes down. If you try, and some people are strong, some people can force a lid to open even though it's not supposed to, that's going to be very dangerous, because what happens is under the pressure the liquid will bubble up at 137 degrees and land on you. So it will come out and destroy you. So you have to be very careful. What we've done to counteract that is most of the benchtop autoclaves now, maybe not all, but most of them have a safety mechanism in place which stops people from opening it. Okay, so it will not open unless the pressure's gone down. So just to bear in mind as you prepare for your projects next semester, you'll bulk up on materials that will need to be autoclaved. Majority of you will. We're seeing it now. There's a nice multidisciplinary spread of material throughout all programs, actually, and that's good to see. So it is possible you'll autoclave materials, but if you're waiting on these guys to be fast, that's not going to work. This is now a 2-hour process because of the safety mechanism. So what might be better for you all, if you're in a bit of a rush, is to bulk them up and put them with the technicians who will put them into the larger autoclaves, and perhaps that would be a faster cycle. But of course you need to do it in the appropriate manner. You don't just run in and dump a lot of material on the technicians and hope for the best. Okay, so I'll describe the process now in a minute. Okay, so this is the prep area in the biology, between the biology labs. So it's between BO8 and BO9, and you're all very familiar with it now. So there's 2 key points I just want to remind everybody of. Is this area here, you're going to see it clustered and filled up with sometimes live cultures, depending on the shelf, and autoclave material then, depending on the shelf also. So the shelves are clearly labelled. If you want to autoclave something, you must put it on the correct shelf with materials to be autoclaved. And if you want to collect your autoclaved materials, they will absolutely be put on the bottom shelf with autoclaved materials labelled by the technicians. If you, by accident, put your materials to be autoclaved on the wrong shelf, they will not be autoclaved because people will assume that's done. It's not a problem so much with this tips and Eppendorfs, because no hazard is really going to happen there other than your material is not sterile. But it is absolutely a problem if you're working with live cultures and you've put them on the wrong shelf and they're assumed to be decontaminated and they're no longer viable, and then the disposal mechanism kicks in then. That's a problem, okay? So you have to be very, very careful where you're leaving your live cultures and be sure to put them on the shelf where they will be autoclaved appropriately, okay? So just bear that in mind. If you're in any way confused, obviously ask. That's really important. And in addition, if you've got plates of cultures, these are the 2 autoclave bins that we have. You always will dispose of your material in plates and tips and so on in the clear bin. Don't go anywhere near the green bin. That's not for you, anybody here, not even for me. What happens with the green bin is after a large batch has been autoclaved, it's put into the green bin to cool down. So don't put fresh contaminated stuff in there. It's not for that purpose. So just use the clear bin and ignore the green bin. Okay? Right. Now, it will probably be a reality, and certainly across the agricultural science, the molecular biologists, and maybe to a degree, but absolutely rarer with the pharmaceutical science, but not impossible. You possibly will be handling live cultures in projects, if not in other classes. And there's mechanisms in place. If you spill them, it's important that you don't just mop it up and dump that material into the bin. The bin is for general waste such as washing your hands and so on. It's not fair to have live cultures on tissues in a bin that a cleaning lady is coming along to take away and dispose of. That is exposing everybody to a live culture, which is not good lab practice, and it's not the way to do things, okay? So if you do spill a liquid culture, you cover the spillage with tissue, and you get that lovely fresh Virkon that I described earlier, and flood it, absolutely, put loads of it all over that tissue and leave it alone. Just leave it there for 15 to 20 minutes and the Virkon will get to work on that live culture on the bench and try and reduce the viable numbers there. After that, then you just mop it all up and you put it into the clear bin that I described down here, so into the clear autoclave bin. Then you go back and you Virkon down your bench, you clean it again with more Virkon and you put those tissues into the autoclave bin. And that's good then. That's pretty much the most you can do, and that's an effective way to get rid of the live culture from the bench. In terms of how you should work around cultures and live cultures, there's a general setup to a bench. So most of you will have this on your project labs, which the onus will be on you, and many of you will have them in general labs. As I said, less so perhaps pharma, but maybe during your project, maybe one or two of you might. So the general rule of thumb is to have your Bunsen close by, have your sterile tips, and have a vessel that will be half filled with Virkon, fresh Virkon, so that you can put your tips as you're working with them into that Virkon, and if there's any live cells on them, they're being decontaminated. So you're not splashing them all over the bench, causing contamination, okay? I think the reality here is that spillages will happen. It's life. You're in college for up to 4 years, and for nobody ever to have spilled a culture in your 4 years of college, well, that's great news. But in reality, they happen. We have measures in place to deal with them, and just be confident as you're working around with those cultures. They're not something to be, I suppose, cautious, overly cautious around. Just respect the nature of the culture that you're working with. Now, with the solid media, some of you will be working with solid plates, and the general rule there is when you're taking from the plate, you only lift the lid from the plate at that point in time when you want to remove material from that solid plate. When you are lifting the lid, you hold the lid in your hand. You never put it down on the bench, okay? So you keep it in your hand, remove what you need to, and put the lid back down. I'm going to be honest, we see a lot of bad practice with that. The automatic, almost reflex for people is to take the lid and put it down on the bench. For two reasons it's bad. Number one, it's causing contamination to the fresh culture you have, okay? So that's bad practice for continuing your work. And then from a safety point of view, you are potentially contaminating your work area and the environment. So if it's a fungal specimen like this guy here, when you lift the lid and leave it wide open, spores will become aerosol, will be available as an aerosol, and they will go and land on other people's project work. And then they'll start probably growing in other people's project work, and then they're getting really unusual results and they can't figure it out. And why is my DNA not even prokaryotic potentially? Why am I getting eukaryote? Because fungal spores have probably started to germinate in there. Or why is my enzyme not giving me the result that I'm expecting from this particular culture? Because it's a different culture growing, and they're now throwing out all of your data. You might not be aware of it. You may not have the, I suppose, maybe the knowledge at that point in time to look for that. And there's 3 weeks' work ruined. Okay, so be careful with that. In general, when you're storing plates as well in the autoclave, one layer of tape is appropriate, but tape them, okay? Because you're going to have a huge number of people coming and going from those incubators, I should say, and they will probably potentially knock loose plates over, and that causes the lid to separate from the plate itself. That's not a nightmare, but they're also possibly going to stick their hands into cultures that they probably shouldn't be. So it's good practice to just put a little bit of tape around stacks of plates and put them in the incubator. Don't err on the caution of trying to totally seal up those plates, okay? Because the base of the plate is loosely fitted into the lid, and it's designed like that for a reason, to allow some sort of air to seep in potentially as well. So if you seal all of that up, you'll probably hinder or impact maybe some growth mechanisms that you're trying to analyze. Also, label your plates appropriately. You never label the cap, you always label the base, the part of the plate that has the agar or the media in it. And the reason for that is you're less likely to knock a base out from under a cap. You're more likely to knock the loose cap off of it, and it's gone missing in the incubator, but at least we know what's in the plate because it's labeled appropriately underneath. Okay, so we know what we're dealing with there. Very important. When you're working with microscopes, remember your slides will need to be disposed of appropriately, and so we use the slide containers that we have left out in all of the labs for you. If you're working with a slide that cracks or breaks because you're heat fixing it, these things happen. They happen more frequently than you would believe. You can't really use that slide for two reasons. Number one, you might not have effectively heat fixed the organism to the slide, or maybe it's a fungal prep and it's not being heat fixed, or it's viable still on that slide. If you continue to try and use it with a microscope and you slice your finger on the broken glass, you could potentially allow entry into your bloodstream of the organism you're dealing with. So that's a bad idea. So you never would work with broken glassware anytime, and especially not broken slides. So you put that slide immediately after it's broken into a clean beaker, fill it up with fresh Virkon, and the technicians will happily deal with that. We have a special broken glass waste disposal for biological slides that have snapped and broken in place. Also, you will probably use stains to analyze microbes under the microscope as well. And I know we always say, put your gloves on. And a lot of people seem to think like, okay, that's because they don't want me to get stain on my hand and it's going to stay on my hand for a week, or they don't want me to, get stain on my lab coat. And yeah, that's one of the reasons. But the reason we want you to wear gloves is because a lot of the stains you use to analyze or see cells under microscopy are a combination of a cohort of different reagents. And some of those reagents are actually nasty and quite toxic, and so you do not want your skin to come into contact with those stains. That's the reason. If it was only because it made your hand blue for the week, we could live with that, but we don't want that to happen continuously. And bear in mind, you do have your lab coat fastened up. Those stains work very well for a reason. So if they get onto clothing, they're staying there. They won't come out of your clothing. So make it be your lab coat that gets stained and not your clothes. Some of you are already familiar with the microscope logs. So in fourth year as well, we'll be issuing a log with all of the microscopes that we hand out. So what does that do? Well, it basically identifies the microscope at that point in time as your responsibility. So you are signing a microscope out. You're going to take responsibility for cleaning it, for storing it appropriately, and for returning it to its storage unit the same way you got it. So that if we pick up on any, misuse of the microscopes, we can trace that back to a particular individual. It hasn't happened yet because the microscope log tends to enforce a sort of an individual to kind of look after the microscope, knowing that they could be called out over it. But what are we looking for here is proper storage of the flex. Do not wrap that flex around the base of the new microscopes. If it cracks, you've got a potential electrical hazard happening. Don't allow excessive oil hang around on the lenses. Clean those lenses down, because if that oil seeps up into the oil immersion lens, it will ruin it, and that will make microscopes few and far between for us because they have to be removed to be repaired. And stains. We now know stains are dangerous. So if there's stain on that microscope, clean it away using the cleaning solution so that you're not exposing other people to hazardous stains. Okay. This is our pathogen lab. Many of you are very familiar with this. We have lots of people around here that work regularly as postgraduates in that pathogen lab. Undergraduate entry into that lab is all but permitted, to be brutally honest with you. Some of you will be doing projects that might require supervised entry into that lab, and you may be handling organisms that are in a particular category, maybe category 2 and 3 organisms as part of your research, less so category 3. I really can't see that happening at undergraduate level, but you may be required supervision in this lab because of the nature of what you're handling as part of your fourth year projects this year. Orla has already mentioned that burns are quite prominent as part of the list of issues or incidents in the department. We have those larger Bunsen burners in place, and just to put it into context, these are the flames. You've seen them already. When we're in labs that have a white background, you're not going to see this high flame over here. So just remember, if you're coming and going from the Bunsen in a fixed period of time, just turn the flame to visible so everybody around you can see there's a huge flame here. I won't reach across that bench for that. Inoculating loop. But if you're going to be moving away from the Bunsen and going elsewhere to do some work, or working on something else for a period of time, just switch it off altogether. It's not a hassle to turn it back on when you need it. But just bear in mind, we do get quite a few burns because of the higher flames that are happening with these newer Bunsens in labs. Okay, lots of you, especially ag science now, that with the changes in their programs, definitely the fourth year molecular biologists, and already the pharmaceutical scientists have already been using these stains. So at various stages across your program, regardless of whether you're doing a project or not on it, in your actual undergraduate modules, you will probably come across these stains, which allow you to visualize DNA or RNA under LED light now, or UV light, depending on where you're analyzing them. So in the past, we would have used ethylium bromide, which was extremely hazardous. We've eliminated that from undergraduate labs, but we will use CyberSafe stain, and in cases we'll use gel red. Now, CyberSafe is almost suggesting that it's 100% safe, and it's actually not. It's reduced mutagenic capabilities and reduced carcinogenicity, but actually it's in a solvent that's called DMSO. And the nature of DMSO actually in itself is it's toxic to mammalian cells, so there's a hazard straight away. And also DMSO actually tends to allow CyberSafe absorb faster through the skin, which is not a good thing, okay? So you still treat it as if it is a hazardous material, because it still is, it's just really reduced. And you would dispose of it as you would your ethidium bromide waste. So you will have designated waste containers on your benches in labs when you're working with it, and you will return all of that waste to the larger waste container in the pilot plant at the end of your lab. When you're dispensing it with your pipette, always check the pipette with IMS at the tip to make sure there's no stain after transferring to the pipette. Sometimes that happens. When that happens, the pipette needs to be decontaminated. So you give that pipette to your supervisor, who will give it to the technician, take it out of circulation, and make sure it's fully decontaminated. Of course, you're going to wear gloves at all times. We know now, stains, wear gloves, no matter if it's DNA or cells. Just keep your gloves refreshed. The difference here is these stains might get on your gloves, okay? So now that glove shouldn't go into general waste. Always transfer your gloves into the designated waste container for any of the nucleic acid stains that you're using, okay? And if in doubt, just put it in there anyway, okay? That's not going to cause any harm. Disposing your materials could potentially cause harm, okay? So always err on the side of caution here. If you spill CyberSafe, the best advice we can offer is that you would try and contain it. So sometimes a tissue, but really the inert absorbent material is the way to go. If you've knocked over a large container, a relatively large container of CyberSafe, stop it from spreading. Mop it up and put it into your designated waste container. IMS has a nice high level of ethanol in there that will wash that stain away from the surface nicely. So continuously wash the surface of the bench with your IMS and put that waste material into the designated waste container. And then together with the technician and the lab supervisor, we'll use a handheld UV light to check the surface of the bench to make sure all of that stain is gone. So it actually can be easily cleaned. It's not something to be awfully concerned about, but just make sure you're gloved up and your lab coats are on when you're using it. Now we have a relatively new imaging system in place. There will be traces of other stains on this system as well. So as a result, it's a touchscreen system. Most of the people here will be using it across all programs. And so we need you to remember to keep your gloves on at all times when you're touching the screen, because that is in use in research, it's in use at undergraduate level and so on. So traces of other stains will be on that. And so people need to wear gloves for safety purposes. It's on a bench now that used to have our older system, which was a different setup. And so we're treating the entire bench, essentially, the large part of it, as a gloves-on region as well, until we're confident that all traces of any stains over the years have been eliminated from that bench. Okay, so gloves on when you're in there. Most of you have already gone through working with acrylamide. Some of you may need to reintroduce yourself to acrylamide as you work with it for a project or a lab. That is also considered separate designated waste disposal chemical or reagent. And so you'll be familiar, you might remember this from some of your second year labs. There is a special waste container for acrylamide use in labs, and the recommendation is that individuals will work on a separate piece of bench coat when they're working with this, so that if a spillage happens, little tiny droplets, you can see it, you can fold up that, and it can go into the waste container there. I chose this as an example, and it's relevant to chemical waste as well. I guess the point I also want to make here is that if you're using glassware with hazardous reagents, your mindset now at fourth year, you're responsible for that glassware. You need to decontaminate the glassware. So not only do you need to know how do I handle it, how do I dispose of it? What do I do if I spill it? How do I clean that glassware now when I am leaving my lab today, so that the technicians can now take it away and just rinse it properly afterwards, okay? So for something like acrylamide waste, it's actually as simple as an awful lot of rinsing with regular water. Just rinse it and then soak in a detergent like Decon 90, and that is good for that glassware there. But you need to be mindful of whatever reagent or chemical you're using, and you're responsible for the decontamination of that glassware. And ask for help, ask for advice, and we're here to help people as well. In your labs, you'll have little tables in all of your manuals now for each of the labs to show you how to handle the waste, how to dispose of the waste. It all might read well and good for some people, and then it might not be entirely clear for other people, and that's not a problem. Make sure you're clear that you're responsible for how you are handling things in that lab, okay? So it's the responsibility. If you're not 100% sure, don't work with something that has a hazard associated with it. Talk to people and be clear on it, and then you're good to go, okay? So there's no problem with that, but this is sort of an example of what you might see. Sometimes you might see MSDS sheets associated with the module in question. They're not up there just for the sake of fleshing out Moodle. They're there because you're expected to read them, okay? Now, of course, you'll get the safety and hazards covered in class, but actually you're also expected to read an MSDS sheet if it's made available to you. At fourth year, you definitely need to be making yourself familiar with them. Those of you that have been in industry already would be very aware of the safety guidelines there and how stringent they are. And those going into industry need to have an appreciation for understanding that as well. Use your safety to your advantage as well. These are, some of the people here might recognise these videos. These are third year assessment videos that I used with my molecular biology students last year. And the expectation is, and will remain to be, that when a hazard is evident, that you will point that out in an assessment or in a written document or in a video, because it's as important as the actual work that you're carrying out. I kept it as anonymous as I could, so we don't know whose hands are there. But you can see what I'm talking about here. You can see the CyberSafe designated waste container. The students have been clever enough to point that out as part of this assessment. That's excellent. You can see here the students have decided to demonstrate how to clean the pipette after use. It's not necessarily 100% relevant to what they're showing, but it's as important as the work they're doing. That's really where we want you to be in fourth year now. Your safety talks. Third year and fourth year students now will always have a pre-safety session before they go into the labs to carry out their projects in pharma and in biology. But we have ag science students and we have agricultural students here as well. And so there's biological safety forms that are very important for you all that are now mandatory as designated by the Health and Safety Committee for all students where relevant. As I said, it's rarer in pharma science, but I'll go through them very briefly. But the point we're trying to make is that none of these students or yourselves will be permitted into a lab unless you know the hazards associated with the materials that you are aware of at that point in time. We understand things will change. That you know the hazard, not a generic term, it's neurotoxin. Like, what does it physically do to you? That's what we need to know. How do you handle it safely? How do you dispose of it? And what do you do if you spill it? Okay, if you know that about any of just the hazardous ones, you know, we're not going to go through every single reagent, you're really confident and you're good to go and you're set up as best as you can be for a safe project. And so you're not now a hazard to anybody else in the lab. That's also very important as well. And like that, you need to talk to your supervisor to make sure you're hitting the nail on the head there with regard to the hazards associated with your sheets. Chemical safety forms, as you all know, are very short. But let's look at the biological safety forms. Anybody swabbing carcasses, you need to fill out a biological safety form. Any swabs coming from agricultural sources, absolutely you need to fill out a safety form. Water samples, you need to fill out a safety form, and so on and so forth. Not only microbiological sources or cell cultures, it's a broader thing we need to consider here. And the reason for that is the sample at that point in time will have little or no potentially no pathogens. And so you're going to culture it and you're going to grow it up to look for a particular organism. But you're not just probably going to grow that one organism up, you're going to grow every single organism up into the millions now. And so it's possible that in that community of organisms that you're growing up, in order to isolate one, there may be an elevation in levels of a certain pathogen there, potentially, just by default. And you need to be careful when you're doing that, and you need to know how to handle it to eliminate that risk, so that you can just get the organism you're isolating or the whatever it is you're interested in, and you don't pose a risk to yourself or other people. So that's where the pathogen lab may come in for some people over the next semester too. The form itself is very brief, it's very easy to fill out, and it's the process of filling it out is very useful to make people aware. Yes, there's chemical hazards, but actually, you know, there's also biological safety considerations for everybody to bear in mind as well. The other point I want to just drive home across the board, there will be audits. I know Orla, our head of department, has already mentioned that. But in addition to the Health and Safety Committee carrying out audits, your supervisor or your programme leader can also carry out audits. So the expectation is that you'll have your safety forms with you in labs. That's the point. They're a summary of the main points that you need to always have at hand when you're working with reagents in the lab. So a small folder with those forms in and signed by both you and your supervisor. And so at any stage, you know, people could be asked to produce those, and maybe, you know, if they're incomplete, that's a nuisance because you're out of the lab, potentially, if you're asked, and that's a day's work gone, which is probably a week's work, and you don't want that to happen, okay? And if we're not happy that people have the safety that they should have before they do their projects. You will be asked to go to the library to bring it up to a standard and before you can commence work. And that's a week's work gone. So, you know, we don't want that. That's not what we're here to do. You're all familiar with this already. You're now in fourth year. We want it to be as smooth as possible for everybody. And that's it. Just enjoy your academic year. Thanks a million.